In this lesson, we're going to be taking a look at the nervous system. The nervous system is a very complex system that is going to be used to respond to different signals and stimuli from other cells and then also from our environment. One way that this is defined is that it is the master control, master communication system for our bodies. And when we're talking about this master control system, there are three basic components to this. There's going to be a sensory receptor, there's going to be an integration center, and then there's also going to be an effector. Between the three of these, there are essentially three overlapping functions. So if we talk about the sensory receptor first, millions of sensory receptors are going to be responsible for monitoring changes that occur both inside and out of our body. So that is called sensory input. So they are going to monitor that sensory input. Then the sensory input is going to be processed and interpreted, and then based on that sensory input, decisions are going to be made. That is called integration. The third part is that a response is going to be affected, and that's going to be by activating muscles or glands via motor output. So three main components. We have the sensory receptor taking the information in, we have the integration system or that integration center, and that is going to process and determine what an appropriate response would be, and then we have the effector, which is going to perform that response. The nervous system itself is going to have a couple different components to it. We have what we call the central nervous system, and that is the brain and the spinal cord. And then we have the peripheral nervous system, which is going to include the cranial and then the spinal nerves. So you can get an overall idea of the different components of the central nervous system and then also the peripheral nervous system looking at this slide right here. Notice that we have sense organs and sensory information that is going to be going into the peripheral nervous system and then up into the central nervous system. And then the response is going to travel back down from the central nervous system to the peripheral nervous system. And then we're going to see the effects in the motor output. So that we have both voluntary, so things that we can control, and then we also have involuntary responses that happen. So again, these two parts of the nervous system, we have the central nervous system and the peripheral nervous system. And when we look at it in this slide, basically the central nervous system is shown as pink, so that is the brain and the spinal cord, and then the peripheral nervous system is going to be the blue part. If we talk about the central nervous system first, this one is going to act as the integrating and command centers of the nervous system. It is going to interpret all the incoming sensory input, and then with that, it's going to issue instructions based on that input. So integrating the information that's received is the main job of the central nervous system. If we're talking about the peripheral nervous system, again, in the image, that is going to be the blue component. This is the spinal nerves, and they will carry impulses to and from the spinal cord. We're going to have cranial nerves. They will carry impulses to and from the brain, and then they are responsible really for linking everything together. So they're going to link all parts of the body to the central nervous system. So notice that the peripheral nervous system is really extending throughout all of the limbs in this diagram. And again, it's connecting everything to that integration center, which is the central nervous system. If we talk about the functional classification of the nervous system, and here we're really talking about the peripheral nervous system structures. We have both afferent and efferent components. So here we have a look at it. When we're talking about sensory input is defined as afferent. You can see that that is basically the guy seeing the glass of water. If we talk about what that is going to do, is that is going to carry the impulses to the central nervous system. So that's the sensory input coming in. And when we talk about that, we have somatic sensory fibers. They carry the impulses from the skin, skeletal muscles, and then also from the joints. And there's also going to be visceral sensory fibers, and they're going to carry impulses from the visceral organs. So that's the sensory input, so things that are coming in and being carried to the central nervous system. Then that is going to be integrated in the central nervous system, and then we have the output part, which we call the efferent part. So this is going to convey impulses from the central nervous system so that we can respond to those impulses. The motor output is going to include somatic nervous system. This is going to allow voluntary control of our muscles. So we will control those muscles and command them really what we want them to do. And then it also includes automatic nervous system, which regulates automatic movements such as the cardiac muscles. So those are things that are involuntary. They are going to happen. We don't have any conscious control of those. The nervous system is composed of two main cell types. The first of these cell types are called supportive cells or supporting cells. And they do have a lot of other names that they go by as well. These are also called neuroglia, glia, and glial cells. And their main job is support, insulation, and protection of the very delicate neurons. Because they're just supporting and protecting, they are not themselves going to be involved in actually transmitting nerve impulses. Now, these supportive cells, there are several different types. So now we'll take a quick look at some of the different types of supportive cells. The most common of the supportive cells are the astrocytes. And the part astra means star, and the shape of these cells is really where that name comes from. So you can see it in the picture here. It does have a lot of long projections from the cell, and those are going to connect to surrounding capillaries and neurons. So these are going to form a living barrier between the capillaries and the neurons. When they do that, they are going to help in determining capillary permeability. They're also going to be playing a role in exchanges between the capillaries and the neurons themselves. Their main job is really to help control the chemical environment in the brain, okay, when they make these connections between the neurons and the capillaries. So that's one type of supporting cell. A second type of supporting cell is what we call the microglial cells. These are going to monitor the health of nearby neurons, and as they monitor the health of these nearby neurons, they're also disposing of debris. And that debris includes things like dead cells and then also bacteria. These cells, the ependymal cells, these are going to contain cilia that beat, and when those cilia beat, remember cilia are projections from the cell, when those cilia beat, they're going to help circulate the cerebrospinal fluid. Okay, so we can see those here, and you can see all the little cilia at the top of them to help circulate that fluid. Other supporting cells, we've got the oligodendrocytes. Those are going to produce fatty insulated coverings. So that's that covering, protection, insulation function of the supporting cells. And then we can also see the satellite cells, the Schwann cells down there in the other picture. Again, these are surrounding the neurons, and important function of surrounding is insulation and protection. Now, in addition to the supporting cells, we do have the neurons. And the neurons, these are going to be highly specialized cells. Their main job is to transmit those nerve impulses. And these are going to contain a cell body, which is going to be a larger portion, kind of big rounded portion of the neuron. And then they're going to have this very long, slender process that's extending from them. And they may have a couple of long processes that are extending from them. So in this image here, the cell body is the part at the top right here where we see that it's rather large. That's where we're going to find the nucleus and a lot of the organelles located at. And then we've got that long process, which is really extending from that point. Here's a closer look at it, the cell body, because that is where we find the nucleus, and it's also where we find the organelles. That is going to be the metabolic center. So that is where the metabolic activities are primarily going to be taking place. The dendrites, which are projecting from that, they're going to be collecting and relaying incoming information to the cell body. So they're bringing the information in, and then the axons are going to relay the information out. So they are sending things from the cell body out of that particular neuron. The axon terminals, okay, which are found at the very end of this long projection, these are going to be ultimately releasing the neurotransmitters, which when they release the neurotransmitters, that is going to be sending the signal out and conveying it to some neighboring neuron, neighboring motor organ. Most nerve fibers are going to be covered with a whitish fatty material, and that is what we call myelin. And in this picture here, you can see that shown in blue. This myelin is going to be protecting, it's going to be insulating, and in the end, it actually increases the transmission rate of the nerve impulses. So by increasing the transmission rate of the nerve impulses, we're going to get a much faster response because of the myelin that's there. So if we go back and we look at the overall structure of the neuron, that actual axon, the part that is projecting from it, so when we're talking about axon, we're talking about all of this here. When we look at that, so remember the axon is for sending the information out. When we look at that axon, notice that it is covered by these cells, which we're calling Schwann cells. And these Schwann cells, they are going to be containing that myelin. So we are protecting the axon, and we're also having the myelin there so that we can get faster traveling of those impulses. If we talk some terminology, first off, tracts. When we use the word tracts, these are going to be bundles of nerve fibers. These bundles of nerve fibers are going to be running throughout the CNS, so that's the central nervous system. The nerves themselves are bundles of nerve fibers, and those bundles of nerve fibers run through the peripheral nervous system, or the PNS. The white matter refers to myelinated regions of the central nervous system, and then the gray matter is going to refer to the non-myelinated regions of the central nervous system. So neurons, when we look at the neurons, they can be classified according to their overall function. So here we see an example of a neuron. Neurons, we can have sensory neurons. The sensory neurons are bringing the information in, so they are carrying the impulses from the sensory receptors to the central nervous system. And then here's an example of different types of sensory neurons. We can have free nerve endings. We might find those in our fingertips. Those are going to be responsible for detecting pain, for detecting different temperatures as well. We have some that are for touch receptors. We have deep pressure receptors, and then we also have motor spindles, and so those are proprioceptors. And so you can see we do have a number of different sensory receptors, which again are collecting information, and then once they get that information, they are sending that sensory input to the central nervous system. So these are all examples of that. Now we also have what are called motor neurons, and the motor neurons are carrying things in the other direction. The motor neurons are carrying information from the central nervous system out and to those responsive muscles and glands. In addition, we have interneurons. The interneurons are also called association neurons. These connect everything together, so they are connecting the motor and the sensory neurons in the neural pathways. Neurons can, in addition to just being classified according to their function, they can be classified based on how many processes they have extending from the cell body. If we look at different ones, we can have what are called multipolar neurons. The multipolar neurons, these have several processes that are extending from the cell body. This is going to include motor and also association neurons, and this is the most common type of neuron that we do have. We can also have bipolar neurons. Notice that with this one, we actually have two processes. We have what we call an axon, and then we also have the dendrite. These are relatively rare in adults, but we do find them in the eyes. We do also have them in the nose. And then this one down here at the bottom is what we call a unipolar neuron. The unipolar neuron is going to contain a single process. That single process is going to very quickly, when it comes out, it's going to split. And when we have that split, it quickly divides into what we call proximal and distal processes. And this is something that we are going to find in the peripheral nervous system. The neurons are going to have two main functional properties. First one is going to be irritability. Irritability is defined as the ability to respond to a stimulus, and when we have that response, they are going to convert that into a nerve impulse. The second functional property is conductivity, and conductivity is defined as the ability to transmit the impulse to other neurons, muscles, and then also glands. If we look at the overall physiology of the nerve impulse, so how everything is going to work, we have a picture here that is showing us what the axon is going to look like. And in this picture, we have a resting or an inactive neuron. Inactive neurons are going to be polarized. And what we mean by that is that there are going to be fewer positive ions on the interface of the plasma membrane, so in the cytoplasm, inside that cell, than we actually have on the outer face, okay, so in the surrounding areas. So notice that it's extra negative on the inside, more positive on the outside. When we get a stimulus, so when that stimulus is received, the permeability of the plasma membrane is going to change. So when we have that change in the permeability, we're going to see that positive ions, specifically sodium ions, are going to start to come into the cell. So they're going to start to enter the cytoplasm, and we can see that happening on the end of the axon here in this image. And when this happens, we get localized depolarization. So by localized depolarization, we start to see an evening out of the charges. There are going to now be more positive ions inside compared to what we have outside the cell. That is then going to start to travel down the axon. So the local depolarization is going to activate the neuron, and when we activate the neuron, that is going to transmit a long distance signal, which we call an action potential. Notice the traveling of that depolarization happening in this particular neuron. The permeability of the plasma membrane is going to change again. This is going to result in blocking of the sodium from entering the cell. So sodium's no longer going to be coming in, and then we're going to get the membrane become permeable to potassium ions. And when it becomes permeable to potassium ions, that means potassium starts to exit. And as the potassium, which has a positive charge, begins to exit, we're going to get repolarization. So this is going to start to reestablish that inactive state. If we look right here, basically we have a sodium-potassium pump. This is a protein that is embedded in the membrane, and its job is going to be to control the input of or the increase of sodium inside the cell, and then it also can send potassium in the other direction. And this pump right here, its job is to restore the original concentrations. So this will restore that polarized neuron, and using ATP. We have to use some ATP to get these concentrations reestablished like they ought to be. Now, many things can inhibit this whole process. So inhibit or hinder the conduction of the impulses. Sedatives can certainly do this. Anesthetics can do this. Low temperatures, so cold, continuous pressure, all of these can inhibit whether or not those impulses are able to be conducted. If we look at the transmission of the signal when we get to the synapse, what we mean by the synapse is this is the tiny little space that is going to be found between neurons. So when we have the impulse actually get to that tiny little synapse, what is going to happen is neurotransmitters are going to be released from vesicles. In this image right here, we can see the vesicles and we see the neurotransmitters as little green dots or circles. And so those neurotransmitters are going to enter that cleft, so the little space between cells. When they enter that space, they will diffuse and travel very rapidly to the nearby neuron. That nearby neuron is going to have special receptors on its surface, which will bind the neurotransmitter. And upon binding that neurotransmitter, that is going to activate some channels, ion channels, that are there at the surface. And when we activate those ion channels, basically open them up, this will cause ions to move in one direction or the other. So this will affect the flowing in of the sodium into the cell, and that will begin that process of depolarization in the next neuron. So that's the physiology of how the signals get transmitted from one nerve to the next. Now, if we talk about the reflexes are going to be rapid, predictable, involuntary responses to stimuli. So basically something that we're not actually going to be consciously controlling. These fall into two categories. We have what are called somatic reflexes. These somatic reflexes are going to be reflexes that stimulate skeletal muscles. The other type of reflex, the autonomic reflex, these are going to regulate the activity of smooth muscles, the heart, and then also the glands. Basic elements of any reflex, and we call it a reflex arc, are going to be 5 different parts to it. We have a receptor, so that receptor is receiving the stimulus, it is then going to transmit that signal to the central nervous system using a sensory neuron. So that's number 2. Number 3 is going to be the integration center. And then we have the motor neuron that is sending the information back out from this integration center, so from the central nervous system. And then number 5 is the effector. So that is what is going to give us that actual reflex or that actual response. Here's just a closer look at it. This is what we call a 2-neuron reflex arc. In this case, you have basically one sensory neuron coming in, carrying the information to the central nervous system, and then we have one motor neuron that's carrying the information back out to the effector organ. Here's a 3-neuron reflex arc. This one is a little bit more complicated. This one you have this extra one, what we call an interneuron, which is connecting the sensory and the motor neurons together. Again, we have information coming in. It is going to go through the central nervous system, and then ultimately it's going to be sent back out to the effector organ.